which is the interface between the external media and the internal media. The supplements which are required for the metabolic activities of the tissues or the cells, like the gases, oxygen, or the water, they pass through the extracellular fluid. And the metabolic waste products which are excreted out of the body, they also pass through the extracellular fluid. So, by maintaining the extracellular fluid volume, kidneys play important role in the homeostasis. Also, in the clinical practice, biochemical investigations are the important weapons for the physicians or the doctors to diagnose the functions and the dysfunctions of the kidneys directly or the indirectly. Renal physiology helps the physicians to know the pathophysiology of renal diseases and plan the management strategy of the disorders. Then we will see the functional anatomy of the kidney. These are primary organs of the excretory system, which consist of two ureters, urinary bladder, and urethra. There is a formation of the urine by the filtration of the plasma, and this urine is excreted through the ureters and stored into the urinary bladder. After the maturation reflex, this urine is expelled out. If we see the gross anatomy of the kidney, these are bean-shaped paired organs located retroperitoneally, size 11 by 6 centimeter in the length, weight 150 grams of each kidney. These are covered by the thick renal capsule. At the medial border of the kidney, there is a concavity through which the ureters comes out. So, these are the parts of the excretory system. Both two kidneys, two ureters, urinary bladders, and urethra. If we see the vertical section of the kidney, it is broadly divided into two regions. Outer part is called as the cortex, inner part is called as the medulla. Outer part, cortex, have the granular appearance, and there is a presence of the glomeruli, then cortical part of the collecting duct, and there is the convoluted tubes. Medullary part is triangular in appearance because of there are the presence of loop of Henle, medullary part of the cortical collecting duct, and the blood vessels. So if we see the vertical section of the kidney, it is grossly divided into two, cortex and the medulla. In the medulla, there are the 8 to 12 conical projections are there. These are called as the pyramids. There are the conical projections are there. These projections are called as the pyramid. And these pyramids are opens into the renal papilla. Renal papilla forms the minor calyx. Minor calyx unite to form the major calyx, and major calyx again opens into the renal calyx. So there are these conical masses in the medullary part of the kidney called as the pyramids, renal pyramid. They open into the renal papilla. They form minor calyx. Minor calyx will join from the major calyx, and major calyx open into the renal calyx. So this is the structure in the vertical section or the sagittal section of the kidney. Then we will see the functions of the kidney. Kidneys play important role in our body. There are the number of functions: secretory, excretory, and the non-excretory functions. First and most important function is the formation of the urine and excretion of the metabolic waste products out of the body. So, by the ultrafiltration from the plasma, there is a formation of the urine. And metabolic waste products like the urea, uric acid, creatinine, these are excreted through the urine out of the body. Second, regulation of the extracellular fluid volume. Extracellular fluid volume is regulated by the secretion of the hormones like the aldosterone, antidiuretic hormone, or the angiotensin. This regulates the extracellular fluid volume and extracellular fluid composition. Next is the regulation of the blood pressure. Regulation of the long-term regulation by the kidneys by secretion of the renin. Renin is secreted from the juxtaglomerular cells of the kidney, and this activates the renin-angiotensin axis. It activates renin-angiotensin axis, and this axis regulates the long-term regulation of the blood pressure. It also regulates the electrolyte composition of the extracellular fluid. Acid-base balance. Maintenance of the acid-base balance by regulation of the bicarbonate excretion and hydrogen ion secretion. It maintains the pH of the blood. It maintains the pH of the body fluids. If there is any abnormality in the acid-base balance, it causes metabolic acidosis or the metabolic alkalosis. So, kidneys regulate the acid-base balance. Second is the regulation of the plasma osmolality by controlling the absorption of the sodium chloride and the water. If there is a change in the plasma osmolality, it is detected by the macular denser cells and cells, and there is a increase there is a regulation of the sodium and the water absorption. Then erythropoiesis. Regulation of the erythropoiesis. Erythropoietin is the main constituent which regulates or which controls the erythropoiesis. Erythropoietin, which is secreted from the kidney. So, if there are the kidney diseases, anemia may be occurred. Kidneys also have the endocrine functions as they secrete the hormones like the prostaglandin and thromboxane. So, these are the functions of the kidney. Then we will see the structure of the nephron. So, what is the nephron? Nephron, these are the structural and the functional, basic structural and functional units of the kidney. There are millions of nephrons are present in the each kidney. They are the hollow tubes. Nephrons are the hollow tubes, and because there is the ultrafiltration or there is a filtration of the blood and formation of the urine. Modified changes are occurs and they convert into the urine. So filtration occurs into these tubules. There are the parts of the nephron. One is the renal corpuscle. Second part is the proximal collecting proximal convoluted tubule. Third is the loop of Henle. Next, distal convoluted tubule. And last is the collecting duct. We will see the one by one parts of the nephron. So this is a picture of the nephron. You will see there is the renal corpuscle or the glomerulus. So this whole structure is called as the renal corpuscle. Then this is the proximal collecting duct. This is the loop of Henle. This is distal convoluted tubule, and this is the collecting duct system. We will see one by one. So this is the renal corpuscle. Renal corpuscle is also called as the Bowman's capsule. It constitutes glomerulus. Glomerulus is the nothing but this is a tuft capillaries. There are the afferent and the efferent arteries. So glomerulus is engaged in the Bowman's capsule, and glomerulus and Bowman's capsule is called as the renal corpuscles. There is a filtration of the plasma and formation of the urine. This takes place through this glomerular capillaries. And urine is called this transport from the Bowman's capsule to this distal collecting duct. This is a transport from the Bowman's capsule to the proximal, from the Bowman's capsule to the proximal collecting duct. So this is the first part of the nephron, that is the renal capillary. Second part is the this proximal collecting duct. It is the initial part of the nephron, so it is called as a proximal. Part of it is a coil, so it is called as a convoluted. So name is a proximal convoluted tube. It is divided into two parts, pars convoluta and pars recta. First 70% part, which is a coil or convoluted, it is called as a pars convoluta. Last straight part, 30% part is the next study, which is a straight, it is called as the pars recta. There are abundant numbers of the mitochondria and epithelial cells are present in this pars convoluta, and they are less in the pars recta. Maximum reabsorption takes place through this pars convoluta part of the proximal collecting duct. Then next is the loop of Henle. This is the loop of Henle. Again, loop of Henle, there are the two parts, descending limb and this is the ascending limb. Descending limb of the loop of Henle, ascending limb of the loop of Henle. Vertical part, there is only the descending limb, there is no ascending limb, but in medullary part, or the juxtamedullary nephron. In the juxtamedullary nephron, there are three parts of the loop of Henle. One is a thin descending limb, thin ascending limb, and thick ascending limb of the loop of Henle. So, this loop of Henle in the juxtamedullary nephron divided into three. There are the three parts: thin descending, thin ascending, and thick ascending loop of the Henle. Then this is the distal convoluted tube. Thick descending limb ends into the distal convoluted tube, and this distal convoluted tube then again ends into the collecting duct. This is the collecting duct. Collecting duct also two parts, vertical portion and the medullary portion. This vertical portion of the collecting duct, and this is the medullary portion of the collecting duct. So same Bowman's capsule, renal capsule also called as a Bowman's capsules, which filters blood from the glomerular capillaries on the alveolar filtrate and delivered into the proximal tubules. Proximal tubule, most part is a coil, so coil convoluted initial segment, so it is called as a proximal. Ends into the loop of Henle, then 15 millimeter long, 40 micrometer in diameter. It is a considerable capacity of the reabsorption. Loop of Henle, vertical part, only descending limb. Juxtamedullary part, there are three parts: thin descending, thin ascending, and the thick ascending limb. Total length 40 millimeter to 26 millimeter. Thin limb 2 to 14 millimeter long. Thick limb 12 millimeter long. 15% of filtrate reabsorbed in the loop of Henle. Loop of Henle of juxtamedullary nephrons plays important role in the counter current mechanism of the urine concentration. Distal tubules 5 millimeter in the length. There are the extensive folding at the basoventral membrane, which provides the luminal surface abundant mitochondria is there. Then it is impermeable to water. Hormones, aldosterone, ADH facilitate its absorption and these segments. Histological picture is different in the different parts of the nephron. In distal convoluted, this proximal convoluted tube, you see this is the histological picture in this part. There are the number of epithelial cells with the number of mitochondria present. In this loop of Henle, the cells are become flat. Again, in the ascending limb of the loop of Henle, there are the less numbers of the mitochondria as compared to this the distal proximal, distal convoluted tube. And in this collecting duct, There are also the flat cells with the less number of the mitochondria. This is the histological picture with the different parts of the nephron, which is important for the absorption of the water as the other substances. There is a difference between the cortical and extramedullary nephron. If we see the location, cortical nephron, superficial region, placed in the superficial region of the cortex, extramedullary in the extramedullary region. Number cortical 85%, extramedullary less, and only 50% are present. Length of loop of Henle in cortical nephron is short, in extramedullary nephron is long. Thin limb in the ascending limb is almost absent in cortical nephron. In extramedullary, both limbs are there present, descending and the ascending. Then tubular function, cortical nephron, filtration and reabsorption. In extramedullary nephron, filtration, reabsorption, and creation of the medullary osmolar gradient. Different arterioles in the cortical nephrons forms the ventricular capillaries. In extramedullary nephrons, they form the ventricular capillaries and also the vasa recta. This is important. There is a formation of the ventricular capillary network, and also there is a formation of the vasa recta in the extramedullary nephrons by these different arterioles. Vasa recta in cortical nephron absent, here is the present. Renin content more in the cortical nephron, less in the extramedullary nephron. Main function, urine formation and excretion of the metabolic waste products by the cortical nephrons, and extramedullary nephrons only there is function in the urine formation. So this is the difference of the cortical nephron and the medullary nephron. This is the cortical nephron. Length of the loop of Henle is small. There is absence of the ascending beginning of the loop of Henle. There is a loop of Henle length is more, and there are both limbs descending and ascending are present. Then we will see the renal corpuscles. The first part of the nephron, that is it. Renal corpuscles consist of the Bowman's capsule, which contains glomerulus and the mesangial cells. So this is the structure of the renal corpuscle, this is the cup-shaped structure. This is a dilated cup-shaped structure of the proximal tubule. And in this there is a glomerulus. The Bowman's capsule consists of the glomerulus and the mesangial. There is a mesangial cells are present here. So this glomerulus is the front of the capillaries, afferent arteriole and the efferent arteriole. It forms the glomerulus. The glomerulus lies this visceral layer, inner layer and the visceral layer. Outer layer is the parietal layer. The space between the visceral layer and parietal layer is called as the Bowman's space. Space is called as the Bowman's space. There is a first step of the urine formation is exhibit in this glomerulus. That is the ultrafiltration. Urine formation is takes place by the three steps. One is the ultrafiltration, second one is the tubular reabsorption, and third one is the tubular secretion. So there are the three steps for the formation of the urine. First one is the ultrafiltration, second one tubular reabsorption, and third one is the tubular secretion. So first step, that is the ultrafiltration of the plasma, this takes place through this glomerular capillaries. And the filtrate which is formed is transported by this Bowman's capsule through the proximal tubes. So this is the renal corpuscles. Initial dilated cup-like structure, layers, visceral, parietal. Visceral layer forms the porocytes of the visceral layer forms the Bowman's capsule. Glomerulus, these are the capillaries, cup of the capillaries are present in the Bowman's capsule. is formed by only capillaries in the body which starts with the arterioles and ends with the arterioles. So, glomerulus which starts with the arterioles and the ends with the arterioles. Glomerular capillaries are the only capillaries in the body that start with the afferent arteriole and end with the efferent arteriole. And these capillaries are covered by the porocytes. These capillaries are covered by the porocytes. And next is the mesenterium. Mesenterium is important component of the renal corpuscle which contains mesenteric cells and mesenteric matrix. These are the mesenteric cells and this is the mesenteric matrix which is also present in the renal corpuscle. Mesenteric cells are derived from the blood monocytes and these are components of the mononuclear phagocyte system. Cells are present in the mesenterium close to the glomerular capillary. This is a glomerular capillary. This is a glomerular capillary. So, these mesenteric cells are present close to the glomerular capillary. If we see the structure of the glomerular capillary, the glomerular capillaries with the fenestrated ends. Where are the fenestrated ends are there? These are the endothelial lining, which forms the filtration barrier. There are the slits or pores are present at the endothelial lining of the capillary, and through which there is a transfer of the molecules takes place, like the glucose, urea, water, these are. But these are negatively charged. These are negatively charged. So proteins are not filtered. So normally proteins are absent in the urine. If there is any dysfunction of this glomerular membrane or the endothelial capillary membrane, then there is a filtration of the proteins, and proteins are of course in the urine, which is called as a albumin. Otherwise, normally proteins are not gases, are not transferred, filtered through this membrane because this is the negatively charged. This is the negatively charged. Then we will see the juxtaglomerular apparatus. There may be short note on the juxtaglomerular apparatus. Draw a level diagram of the juxtaglomerular apparatus and this is that. So, if we see the structure of the juxtaglomerular apparatus, this is the location of the juxtaglomerular apparatus. When thick ascending wing of leukophilate, when thick ascending wing of the leukophilate comes in contact with the glomerulus of the same renal corpuscle. This thick ascending wing of leukophilate comes in contact with the glomerulus of the same renal corpuscles. There are the modifications, tubular modifications are occurred. There, tubular modifications are occurred into this thick ascending wing, and also there are the modifications occurred in the afferent and efferent arteries of the glomerulus. This whole modified structure is called as the juxtaglomerular apparatus. This whole modified structure is called as the juxtaglomerular apparatus. Say what I mean again, when the thick ascending wing of the leukophilate comes in contact with the glomerulus of the same renal corpuscle, tubular modifications are occurred. There are modifications in the cells of this limb, and also there is a change in the modifications in the afferent and efferent arteries. And this whole modified structure is called as a juxtaglomerular apparatus. See, thick ascending wing of leukophilate comes in contact with glomerulus of the same renal corpuscle. Structural modifications occur in the tubule and afferent and efferent arterioles. Entire modified structure is called as a juxtaglomerular apparatus. It includes macular-dense cells of the thick ascending limb, juxtaglomerular cells, and gaseous cells of the mesenteric cells. There are the three parts of the juxtaglomeralular apparatus: macula densa cells, juxtaglomerular cells, and the thin assembly. So this is the structure of the juxtaglomerular apparatus. The macula densa cells. These are the cells of the distal convoluted tubule. These are the cells of the distal convoluted tubule. Histological changes are occurs in these tubules. The cells are densely arranged, so these are called as the macula densa cells. These are very sensitive to the changes in the axial composition and axial quality. Extracellular fluid change in the volume or the composition. These are sensed by the macula densa cells. They act as a chemoreceptors. Macula densa cells act as the chemoreceptors. So these are the histologically changed cells of the distal convoluted tubule. Densely arranged called as macula densa cells. They are sensitive for the change in the extracellular fluid volume and extracellular composition. These are also called as chemoreceptors. They act as a chemoreceptors. Next cells are the juxtaglomerular cells. Juxtaglomerular cells. These cells are at the afferent artery. These are the juxtaglomerular cells, where there are the modifications that takes place in the afferent arterial cells. And these juxtaglomerular cells secretes renin. Okay, juxtaglomerular cells secretes the renin, and which activates the renin-angiotensin axis, and which is responsible for the regulation of the blood volume and the blood pressure. Next cells are the mesangial cells or the Lassus cells. These are the mesangial cells or the Lassus cells. The angular part, which is made by afferent and inferior artery, this angular portion, which is a form because of the afferent arteriole and the efferent arteriole, and there the cells are present. These cells are called as the messenger cells or the Lassus cells. They are also called as the extraglomerular cells. These cells are called as the messenger cells of the Lassus cells. Then these are the formous intramural capillaries are present in the movement space. Endothelial language of the glomerular capillaries. So this is the structure of the glomerular juxtaglomerular capillaries. Macula densa cells, modified epithelial cells. Densely arranged, so it is called as a macula densa cells. Act as a sensor, monitors change in the ionic composition and rate of flow of the tubular fluid. Provides appropriate feedback signal to the renal corpuscle to change the rate of the filtration. It is an important component to the tubuloglomerular feedback circuit. Juxtaglomerular cells, granular epithelial cells, present in the tunica media of the afferent arterioles. These granular cells are modified as per smooth muscle cells with epithelial appearance, contains secretory granules, secretes renin, which activates renin-angiotensin system. Secretes renin, which activates the renin-angiotensin system. Lassus cells or messenger cells present in the triangular space formed by the afferent and efferent arterioles and the macula densa. These are messenger cells present outside the glomerula, also called as extraglomerular messenger cells. Secrete some quantity of the renin and erythropoietin. That is the function of the Lassus cells. They secrete renin and the erythropoietin. So, what are the functions of the juxtaglomerular apparatus? Juxtaglomerular cells secretes the renin, which involved in the activation of the renin-angiotensin system and regulates the blood volume and the blood pressure. Second, macula densa cells, which acts as the sensor, which detects the change in the rate of flow and volume of the flow in the tubule. Third, Lassus cells or the messenger cells secretes the renin and the erythropoietin. Erythropoietin, which is a main for the erythropoiesis. So these are the functions of the extracellular apparatus. Then you will see the renin-angiotensin system. Renin is acid protease secreted from the extracellular cells. Is a glycoprotein with a molecular weight 37,326. It first synthesized as the pre-prorenin, which converts into the prorenin and finally becomes the renin. Renin acts as an enzyme which converts angiotensinogen into the angiotensin 1. It acts as an enzyme which converts angiotensinogen to the angiotensin 1. So see the renin which is secreted from the extracellular cells which converts angiotensinogen which is secreted by the liver into the angiotensin 1. Renin secretion is regulated by regulation of the extracellular volume, blood volume, and the blood pressure. It activates renin-angiotensin system, and then it regulates the tissue volume, blood volume, and the blood pressure. Factors which increase renin secretion, decrease blood volume, or decrease of blood pressure. Decreased blood volume or the decreased blood pressure causes increase in the renin secretion. Decrease concentration of the sodium in the plasma or increase the concentration of the potassium causes the increased renin secretion. Increase sympathetic activity, increase renin secretion, increase circulating catecholamines causes increase renin secretion. Prostaglandins increase the renin secretion. Factors decrease renin secretion are increased sodium concentration, then increased blood pressure, antidiuretic hormone, and angiotensin 2. These all decreases the renin secretion. The conditions where renin secretion is more. One is a hypotension, second, decreased sodium concentration, third, increased calcium concentration, potassium concentration, hypovolemia, dehydration, renal artery stenosis, cardiac failure, in standing posture, cirrhosis of liver, or psychological depression. These are the conditions where renal secretion remains high. So, this is a renin-angiotensin system. Liver cells secretes the angiotensinogen. This is a formed by the liver. Angiotensinogen is in the inactive form. There is no any effect of this angiotensin on the blood volume or blood pressure. Renin secreted by the extracellular cells when there is a hypovolemia, when there is hemorrhage, decrease the blood pressure or decrease the sodium concentration. Because of this hypovolemia, hemorrhage, hypotension or hyperlipemia, extracellular cells secretes the renin. This renin acts on the angiotensinogen and converts it into the angiotensin 1. Converts into the angiotensin 1. Angiotensin 1 is also the inactive form. This angiotensin 1 is converted by angiotensin converting enzyme, which is a secreted from vascular endothelial cells of the pulmonary blood vessels. This angiotensin converting enzyme is secreted by the endothelial cells of the pulmonary blood vessels, and it converts angiotensin 1 into the angiotensin 2, and this angiotensin 2 is an active form. This angiotensin 2 causes vasoconstriction, aldosterone secretion, salt and water reaction. Angiotensin 2. Because of the vasoconstriction, there is increase in the blood pressure. Aldosterone secretion, there is increase in the blood volume. sodium and water reabsorption also there is increase in the blood. This angiotensin 2 is again converted into the angiotensin 3 by peptidases, and this angiotensin 3 is also activated form which causes progesterone secretion and the vasoconstriction. So again, the vasoconstriction and progesterone secretion causes increase in the blood volume and increase in the blood pressure. Angiotensin 3 converts into angiotensin 4 and it reactivates metabolites. So only two active forms are there, angiotensin 2 and angiotensin 2, which causes this effect, and because of this vasoconstriction or sodium and water reabsorption, there is a increase of blood volume and increase in the blood pressure. Thus, the renin-angiotensin system is acts. So there is the activation of the renin-angiotensin system. What is the clinical importance of the renin-angiotensin system? It is implicated in the genesis of the hypertension. Renin increases angiotensin, which causes vasoconstriction, and because of vasoconstriction, there is a raise in the blood pressure. Renin secretion is more when blood pressure is more. It is important physiological basis of the renal hypertension. It is the renin-angiotensin system is physiological basis for the renal hypertension. Etiologically, there are the hyporenin hypertension, normorenin hypertension, and hyporenin hypertension. There are the three types: hyporenin, normorenin, and hyporenin hypertension. Treatment for the hypertension with the drugs which inhibits the this anticholinesterase blockers, ACE blockers, drugs which inhibit ACE blockers effectively controlling the blood pressure. These are the captopril and enalapril. Also, angiotensin 2 receptor blockers are the losartan. So these are the treatment for the hypertension, that is the captopril, enalapril, and the losartan. Then renal circulation. You will see the blood supply to the kidneys and about the renal circulation of the kidneys. Kidneys receive 23.5% of the cardiac output. Blood flow to the kidneys is 1,260 ml per minute. More amount of the blood supply to the kidney as compared to the other tissues in the body. Or 420 ml per 100 gram of the tissue per minute. It is more than the cardiac tissue supply. There is a more abundant number of the blood supply to the kidneys. But it is not for the metabolic need, but it is for the filtration of the plasma. It is for the filtration of the plasma. So there is the kidney blood flow is more. That is 1,260 ml per minute or 450 ml per 100 gram of the tissue per minute. It is maximum in the cortex and less in the medulla. Average blood flow in cortex is 5 ml per minute per gram. In medulla, only 0.5 ml per minute per gram. Low blood flow to the medulla, it maintains hyperosmolality in the medullary interstitium. Because of this low blood flow, it maintains hyperosmolality in the medullary interstitium, and which play important role in the counter current mechanism of urine concentration. These play important mechanism, that is the counter current mechanism of the urine concentration because of the hyperosmolality in the medullary interstitium. And this hyperosmolality is because of the low blood flow to the medulla, that is only 0.5 ml per minute per gram. If we see the arterial supply to the kidney, each kidney there is a single renal artery which originates from the aorta. Renal artery branches to form segmental arteries. Segmental arteries form interlobar artery. which form arterial artery, interlobular artery, afferent artery. Afferent artery to the glomerular capillary network, that is glomerulus, forms the efferent artery. Efferent artery lead to the second capillary network, which is formed around the renal tubules, that is a peripheral capillaries. So this is the blood supply to the kidney. See, this is the arterial artery, which is a forms into this the interlobular artery, and interlobular artery forms afferent artery. Afferent artery forms the efferent artery. This efferent artery peripheral network. This is peripheral network, and it's called as peripheral capillaries, and they forms the vasa recta. It forms efferent arteries forms the vasa recta. Vasa recta in the middle portion of the extracellular network. So, efferent artery forms peripheral capillaries and also makes the formation of the vasa recta. So, this is the blood supply to the kidney. Again, renal artery, renal artery, segmental arteries, interlobar artery, arterial arteries, interlobular arteries, to the afferent artery. Afferent artery to the glomerular capillaries. Forms efferent arterioles, peripheral capillaries or the vasa recta, interlobular veins, arterial veins, interlobar veins, segmental veins, and the veins. You see the picture again. So this is the vasa recta. This is the formation of this interlobular vein and the blue color is the vein. So this is the blood supply to the kidney. What is the importance of the renal blood flow? It supplies oxygen, nutrients, and hormones, delivers metabolites and waste products of the kidney for excretion in the urine, controls concentration and dilution of the urine, influences solute and water reabsorption from the kidney, determines glomerular filtration rate. So these are the importance of the renal blood. Regulation of the renal blood flow by the three factors: neural factors, hormonal factors, and local factors. So first we will see the neural factors. That is the sympathetic control. Stimulation of the renal sympathetic fibers causes vasoconstriction, which decrease the blood flow to the kidneys, and conditions activate renal sympathetic activities are hemorrhage, cold, pain, exercise, anesthesia, and this decreases the blood flow to the kidney. Sympathetic stimulation increases renin secretion from juxtaglomerular cells, activate renin-angiotensin system. Formation of angiotensin 2 causes renal vasoconstriction, decreases the blood flow. Angiotensin 2 and sympathetic stimulation of the kidney stimulate production of the local prostaglandin, produce vasodilatation, and oppose the vasoconstriction. Hormonal factors, circulating catecholamines, and vasoactive peptides like angiotensin 2 cause renal vasoconstriction. Dopamine in high dose causes renal vasodilation, therefore dopamine without positive effect of carbohydrate. Local factors, locally produced metabolites and chemicals like carbon dioxide, prostaglandins cause renal vasodilation and maintain renal blood flow. So adenosine is a potent vasodilator in coronary and cerebral circulations. It causes constriction in the renal artery. See here, adenosine is a vasodilator there in the coronary circulation and in the cerebral circulation. But it causes vasoconstriction in the renal circulation. Autoregulation, blood flow to kidney maintained at the constant level despite the change in the systemic blood pressure between 80 to 180 millimeter of Hg is called as the autoregulation of the renal blood. Metabolic mechanism contributes autoregulation of the blood flow. Nitrous oxide, prostaglandins, inosine, and other metabolites influences the blood. Glomerular feedback mechanism plays a role in autoregulation of the blood flow, in which composition and flow of physical fluid determine red blood flow and glomerular filtration. So this is about the renal circulation of the renal blood.